Hello, everyone. Thank you very much, Gloria. Thank you for organizers. I'm Anton. It's my great pleasure to be here today and present. I'm an ETH fellow and postdoctoral researcher at the Food Systems Biotechnology Group at ETH Zurich in Switzerland, where I worked on the Microbiota Vault Initiative. And the Microbiota Vault has just concluded its launch phase, as Gloria mentioned in her talk, and which really required navigating very complex local and also global regulations and technical challenges, but also opportunities to really share the future, to really shape the future of this truly global mission, as we have now just seen from Gloria's talk. So in the launch phase, we were focusing on the development of standardized protocols for sample collection, preservation, transport, metadata development, and data annotation, each of which probably deserve a separate dedicated talk. But to give you an overview of some of these activities in my talk today, I'll focus primarily on data annotation and current collections we have in the Microbiota Vault. So while really refining the working model in the launch phase, trying to understand how this initiative can be viable and work well, we're focused on two sample types. As Gloria mentioned in her talk, we plan to scale in the future, but the first two sample types were human stool and fermented food. So now it's very unlikely that today I need to convince anyone in the audience that the gut microbiome is important for human health, but so are the fermented foods. And this is mainly because they make up about 1/3 of foods consumed globally, and they're primary natural source of microbes in human diet. And those microbes, they confer potential health benefits, and especially for gut health. And I guess in the context of Microbiota Vault, what is really important is that they are human culture associated foods, and microbial biodiversity with them are human culture associated microbial biodiversity. And although fundamentally two different sample types, they share a similar fate with human gut microbes, as in that they are vulnerable to industrialization. So in the launch phase, we established a number of international collaborations with local working collections who were interested in depositing both sample types into the Microbiota Vault. And we focused on developing minimal metadata requirements that would provide contextual information for these samples. And the goal here was really to find a sweet spot between what is feasible to ask from these people and what makes for a comprehensive report. And after all, metadata really provides the key to gaining biological insight from any microbiome data or physical specimens. So building on existing community-driven standard, we developed a series of metadata templates with a unified vocabulary to ensure consistent and comprehensive metadata across collections. And we also developed a comprehensive user guide and offer support and metadata validation service to our partners who were required to complete the metadata prior to depositing their specimens to the Microbiota Vault. So to give you a bit of a flavor for the minimal human metadata, we require 21 fields that capture information on sample identifiers, environmental context, various spatio-temporal information, as well as technical notes, for example, on cold chain during the sample transit that ensures the sample quality, and also sample ownership and provenance information of the depositors. For the food samples, this also include a number of food-specific metadata fields that would capture information important for the food samples, such as food source, food ingredients, and for example, local traditional name of the foods. When the samples were deposited to the Microbiota Vault and arrived in Zurich, and if we were authorized by the depositors who maintain ownership of the samples to sequence these samples, we then process these samples using standardized protocols such as those developed by the Earth Microbiome Project to generate amplicon sequencing data. We also developed and tested analytical pipelines, and we distributed these pipelines that can be used by the Microbiota Vault in the future, but also local working collections and their collaborators, for example, to characterize microbiota in the deposited samples. So now, with all these efforts behind us, I want to provide a quick overview of the current collections deposited to the Microbiota Vault and the outcomes of the launch phase. We initiated these collaborations with multiple collections who deposited human-associated samples. There were fermented food samples from Benin, Brazil, Ethiopia, Ghana, Laos, Thailand, and Switzerland. And in total, nearly 1,200 human stool samples and almost 200 fermented foods. We processed these samples, as I mentioned, using standard methods and generated high-quality amplicon sequencing data for nearly 850 human gut microbiome samples out of these 5 local working collections. And we also used food-specific protocols to process fermented food samples and generated amplicon data for nearly 170 samples from 3 collections, which includes animal-based foods and plant-based foods. Some examples here include, for instance, fermented mangoes, rice noodles, and fish from the rivering communities in Thailand. So these data sets are now shared with the local working collections who can pursue in addressing their own research questions using this data, and the samples are now safely stored in a biobanking facility in Zurich. The launch phase was a truly team effort led by scientists from research groups of Pascal Van Esch, Nicholas Bakulich, and Adrian Egli here in Switzerland, with support from key foundations. So now, as we hope that these samples are here to stay, we want to ensure that they maintain viability and they're suitable for recovery and potential future applications. So we also performed the long-term cryopreservation validation experiment, and my colleague Youzheng Teo of the University of Lausanne will share the outcomes of this work in his talk. With this, I'd like to thank you for your attention, and Youzheng, the virtual stage is yours.
Thank you. Thank you, Anton and Gloria, for introducing the topic and also leading so nicely into my topic. So I'm Youzheng Teo. I'm from Pascal's Von Esch lab at the University of Lausanne, and we're part of the Microbiota Vault launch phase. So I'll talk about one of the goals of the Microbiota Vault launch phase, which was the validation of the long-term cryopreservation efficacy for viable microbial recovery. So I will start with a little bit of background and why is it important. We all know that the gut microbiome is essential to human health. However, most research today is dominated by DNA sequencing rather than work with live bacteria. And cultivation of live bacteria from complex communities such as fecal samples are extremely crucial for mechanistic studies and development of microbiota-based therapeutics. However, effective long-term preservation strategies focusing on maintaining microbial viability and community recovery are still poorly understood today. Therefore, in our current work, we aim to evaluate the protective efficacy of 8 different combinations of cryopreservative treatments, and this is done under 2 different storage conditions across different groups of infants, adults, and children. And this is due to their different microbiota makeup. And all this is done with a focus of implementation in a low resource setting. So to understand how we aim to achieve this, I will start with an overview of what we have done so far. So we first started with the recruitment and collection of fecal samples from adults, children, and the infant groups. The samples were collected and sealed within an anaerobic pouch, and they were either picked up directly or shipped overnight by post. Upon receiving the samples, we then processed the samples immediately and took apart a sample for directly sequencing of the fresh stool samples. We then also directly plated the stool samples on 6 different types of rich media as listed below. And this would give us an idea of what we are able to recover and grow on day 1. We then subjected the samples to 8 different cryopreservative treatments, and these are also listed below. And afterwards, we stored them for a year in liquid nitrogen or -80. And after a year of storage, we then plated them. And to characterize the microbiome profile, we then subjected them to DNA extraction and then further pooling of samples from different plates, from the different rich media plates, and then performing 16S amplicon sequencing. So this then allows us to compare A, what is the original microbiota composition, versus B, the fraction of viable microbiota that we're able to grow and recover, and C, the same after a year of storage. So what do we learn from the sequencing data? We can first start by looking at the microbial diversity across the different cryopreservative treatments. And unsurprisingly, we see that the fresh stool sample had the highest alpha diversity among all of them. What was surprising was that we see that the alpha diversity across all different preservatives were extremely similar compared to the direct plating and the rest. What we also see is that traditional glycerol-based preservation and snap freezing are equally as effective as more complex and costly treatments. We then also looked at the two different storage conditions that we tested, the -80 and the liquid nitrogen, and here we did not see any significant difference between them. Next, we then looked into the microbial composition of each sample across the different treatments. And we saw that there's a substantial overlap of 122 genera, and this accounts for over 99% of the total relative abundance. And this is looking at the overlap between the fresh, the directly plated, and all the different preservative treatments. And only 27 out of the 227 genera were unique to the fresh stool sample, and this only represented 0.02% of the total relative abundance. What was next to further characterize the difference of the fecal microbiota composition between the different samples, we then performed a unweighted unifrac analysis, looking at their different beta diversities. However, it appears that our sample seems to exhibit a subject-specific clustering rather than by age or different treatment groups. To reinforce this, we then perform a random forest classification, which shows that the sample again exhibits strong inter-individual identity, where the culture samples after a year of storage are often correctly predicted with 98% accuracy back to their subjects or fresh stool samples. Lastly, we looked at the differential abundance of bacterial genera across different preservative treatments, separated by the different age groups. And here in green, we see the species that has been enriched, and in brown, the species that has been depleted. However, overall, we see there's a depletion of the strict anaerobic taxa, such as Anaerostypis, Intestinibacter, Ruminococcacea, and Eubacterium after a year of storage. In addition, we also saw depletion of taxa such as Akkermansia species, and these are likely due to their restrictive dietary niches. So in conclusion, I hope that from my presentation, I told you that, or you got that conventional glycerol-based preservation and snap freezing are comparable in performance to more elaborate and costly protocols, which are more favored in low resource settings. There is a lack of strong treatment-specific effects on microbiota composition, and this suggests a shared response towards freeze-thaw stress, and this in terms then favors fast-growing taxas. Also, lastly, there's intrinsic properties of the fecal matrix appears to be able to preserve bacteria to a certain degree. Next, we had some limitations and outlooks of our studies. The first being that our study had a limited sample size and was based in Switzerland, and this led to lack of global representation. Second, our studies focus mainly on the bacterial communities, whereas there's other microbes such as fungi or viruses that may provide important functions or contribute to our human health. Lastly, a follow-up of further evaluation of long-term cryopreservatives are needed to study, for example, more than 5 years up to 10 years to really allow for study of long-term storage effects. And to conclude, for the Microbiota Vault launch phase, we would like to again remind that we were able to achieve that we were able to establish a biobanking infrastructure in Switzerland, and this is located in the Institute of Medical Microbiology at the University of Zurich. We have also established standard operating procedures for sample collection, transport, metadata reporting for both human stool and fermented food. Lastly, we were able to set up 9 local working collections around the world, 5 collections focusing on gut microbiome and 4 on fermented food. And so far, 128 million unique sequences and metadata are to be published under the Creative Commons license with attributions to the local working collections. And this sets us off on a good foot for phase 2 of the Microbiota Vault. So for acknowledgement, I'd like to thank my lab and all the members involved in the launch phase, and also the organizers for allowing us the opportunity to present.